everyone. Welcome to the contributed talk 3. And today we'll have 4 talks on this session, and they're about predictive coding and auditory cortical neurons of songbirds, model connectivity, overcoming limitations of functional connectivity by leveraging the power of VOXELWISE encoding model framework, and we'll learn about model METAMERS, how they illuminate the divergence between biological and artificial neural networks, and finally, how Go distort the representation of space. So let's just get started, and let's welcome Srihita to learn about predictive coding and auditory cortical neurons of songbirds.
Okay, hi everyone. Thank you to CCN for organizing this. Today I'm going to talk about some of our work on predictive coding and auditory cortical neurons of songbirds. Okay, let's start with an example. Suppose I'm coming to this conference, so I want to search something on Google about San Francisco. So Google throws me a few suggestions. Initially, I start typing in San, and then it thinks I'm in San Diego because that's where I'm from. And then most probably because of me searching about local news and everything. But as I type in San Francisco, it throws me a variety of suggestions, mostly complaints about the weather or how expensive the Bay Area is. So something very similar happens in our brain. So when we perceive information in the sensory world, we make inferences based on what is already known. So the idea of perceptual inference, more so for temporally patterned signals such as speech and audio, is a dynamic process that unfolds across time. So this inferential process is tied to an underlying generative model that combines information across sensory events and helps to organize this complexity of the natural world. So one theoretical framework within which the kind of inferential knowledge might be imparted to perception is called predictive coding. So in our study, we wanted to test specific hypotheses in the auditory system about the relationship between individual neurons and the extent to which they carry information either about this generative model, that is prediction, or about the error that might arise from the prediction and the incoming sensory information. So in short, we wanted to test if there was evidence for this predictive coding hypothesis in single neurons with the assumption that either the prediction or the error is encoded in these neurons. Okay, so qualitatively, we've seen evidence for this hypothesis across sensory modalities. Here, we're going to study this phenomenon in songbirds, especially the European starling. And the reason we're using songbirds is because they can produce these learned spectrotemporally rich songs in the course of their natural behavior that are temporally organized. And predictive coding offers an excellent framework for studying temporal pattern signals, such as speech. In addition, it also provides the opportunity for investigation of song learning and vocal communication. So to study this phenomenon directly in starlings, we can split it into 2 parts. In part 1, we will introduce a framework to obtain 3 different representations of the stimulus guided by the predictive coding framework, that is the signal, the prediction, and the error. And in part 2, we try to map neural responses onto each of these different representations. Okay. Again, to reiterate, for temporally patterned signals, we consider the idea of predictive coding as sort of the prediction of the future in your time series. So for the first part, we built an explicit model of this prediction where we take past events and build a representation of what is expected to happen in the future. So to do this, we first take waveforms of natural European starling birdsong, and then we Fourier transform them to convert into spectrograms. We then segment them into pass segments of about 170 milliseconds, and the next immediate time bin as future segments of about 10.5 milliseconds. We chose the length of these pass segments based on the average length of like stalling motifs, so at least like there was a previous knowledge of at least one motif. And then these are then sent into the temporal prediction model, or TPM, where we have the past segments as inputs, and we try to predict the future segments. So here I have the architecture of the model. The TPM is a simple single-layer feed-forward network model. The inputs here are the past segments. These are linearly mapped onto the hidden layer or the latent layer through weighted connections, which is then followed by a sigmoid nonlinear transformation to predict the future, the true future. In our case, that was the next time in. So we trained the TPM on an extensive corpora of birdsong spectrograms to generate a latent predictive feature space that comprised of 256 units. Under this model, the past spectrogram segment contains a more generalized class of spectrotemporal features of the incoming song. So that was the signal component of the framework. The latent space that contained predictive spectrotemporal features of song represents the prediction component, and the prediction error component of the framework is captured by computing the difference between the output of the model and the true song. Okay, we then just resized everything to be of the same dimensionality for our next step. Here we have some examples of the acoustic feature spaces that have been outputted by our model. Here in the first row I have the spectrotemporal features, 5 examples of them. The second row is corresponding predictive features, or the TPM features. And the third row are the corresponding error features. The sensory system has a hierarchical organization. This means that different regions might potentially express different components of predictive coding in different ways. Our regions of interest include NCM, CM, and field L. The reason we are recording from field L is because it's an analog of the human primary auditory cortex, and field L directly gets projections from the midbrain and the thalamus. And then it sends projections to CM and NCM. Both are thought to be analogs of higher order auditory cortex in humans. So these recordings were done in anesthetized birds, and later I'll talk a little bit of data from like awake birds. But here on the right, I have spectrogram segment of an acoustic stimulus that is composed of segments of this natural starling song, and then I have its corresponding spiking response in NCM. So the second row is a spike raster of a single neuron with multiple representations of the stimulus above. And then in the third row, we have a response of a population of about 150 units. As we can clearly see, NCM cells typically show these sparse, highly selective responses to natural birdsong. To relate these acoustic spectrograms to neural activity, we then computed receptive fields using the maximum noise entropy or the M&E model. So previous work has shown that the M&E model well describes the sensitivity to higher order features that are exhibited by these NCM cells. So this approach seeks the most random or highest entropy model that is consistent with known stimulus-response relationships. So for the optimization of this model, we used 80% of our spectrograms for training and the rest for testing. The optimal model yields this logistic function. The reason it is logistic is because spiking response is binary, and it has parameters A, H, and J. The parameters constrain known stimulus response relationship. A constrains mean firing rate, H constrains spike triggered average, and J constrained spike triggered covariance. So then for each unit, we independently computed these optimal parameters A, H, and J for the M&E model, and in which the stimulus was either the short-time Fourier transform spectrogram, or the FFT, or the stimulus was the projection of the spectrogram into the TPM latent space, or it was the error between the future spectrogram outputted by the TPM and the true future spectrograms. So this yielded a version of each neuron's composite receptor field that was fit to either the TPM in the first row. So point to note here that you can see spectral features in the FFT receptor field, or in the second row it was fit to the TPM features of song. The TPM features don't have a time frequency organization because it's only like a latent weight matrix, and that's why it looks noisy. And the third row are the spectral temporal features of the error, of the prediction error. So the linear feature, or the first column, corresponds to the H parameter of the model. And then on the second column, we have the eigenvalue spectrum of the J matrix. So the eigenvectors of the extreme points on this spectrum represent the most significant features. Here I have excitatory features which were associated with negative eigenvalues. Likewise, the inhibitory features are associated with the two most positive eigenvalues. So we expect the features for, as you can see in the third row, we expect the features for the error representations to have some kind of spectral organization, but they're less obvious than the FFTs. That is because this has been filtered through our model. So the features of the trained M&E models now that we obtained, like AH and J, these can be used to predict spiking response of a neuron to novel stimuli. Here I have an example. So here the black curve shows the firing probability outputted by the M&E to the stimulus segment in the top. The red curve shows the empirical response of the cell. So we evaluated the model by measuring Pearson's correlation coefficients. In this example, as you can see, the model seems to do really well, and the coefficients can get pretty high as 0.76. So now what we can do with these trained parameters is we could plot the correlations between the modeled and the actual responses when the M&E is fitted to all the three stimulus representations. Here in this first plot, we have like the percent of response variance, which is square of our correlation coefficients that can be explained by either the FFT, the TPM, or the error. In NCM, the TPM explained over 40% of the variance, while the error only explained 8%. However, in CM and FieldL, the error representations outperformed those in NCM to explain about 55% of the variance. We can also see an interaction between like the representations and the nuclei in CM and FieldL. What another point that we should note here is that we know that we can drive this M&E model with the signal or the FFT. We have also shown in the previous examples that we can get really high correlation coefficients, and that is why you see such a high explained variance percent for the FFT, but that does not mean that the other representations are not captured by the spike pattern. The FFT is in fact a function of the TPM and the error, and the real question is how much of that can be attributed to the prediction versus an error between the prediction and the signal. Next, we wanted to see if the neurons can be categorized into prediction or error neurons in some way. So for each unit, we assigned the response at all time points to either the prediction or the error, depending on which one is more likely to drive the response at that point. As we can see, again, NCM individual neurons most distinctively appear to be carrying information about predictive features in their response. And CM and FieldL neurons lean towards the negative side, or the error neurons. However, they are not as distinctive as NCM, and they seem to share a lot of variance with prediction. For future directions, we wanted to record from awake behaving subjects. Here we have a pilot study from one bird. When the animals are awake, qualitatively they look very similar to the anesthetized responses. The general properties appear to hold, although quantitatively there might be some important difference that we're currently investigating. So, in summary, there is solid evidence for predictive coding in the auditory system that's consistent with all of the frameworks that have been proposed. So, NCM individual neurons most distinctively appear to be carrying information about predictive features in their response. Alternatively, CM and field-held responses are better represented by acoustic features that can carry information about the prediction error component. Although there seems to be a high degree of shared variance with the prediction in comparison to the NCM. So in this study, we have proposed a framework to test predictive coding hypothesis, and we showed that with only this one architecture here, that is our feed-forward model. We chose this model because the model resembles the linear, nonlinear cascade of sensory neuron firing. But in the future, there are a lot of questions to ask if a range of architectures for the generative model can be optimized for different regions, because we might expect field L to have a different reorganization than the NCM or CM. And we could also change the delay to prediction. Here, I just use the next immediate time bin and the size of the network and parameters to see how this organization changes. That's all. Thank you. I want to thank my lab, Tim, for being a wonderful PI, and Brad and Michael for helping me collect all this bird data, and the rest of Gendra Lab. And if you want to get in contact with me, I have my information there. And thank you. Thank you for CNN organizing this.
Cool, thank you. Yeah, please go ahead.
Hi.
Hi.
Oh, thank you for the talk. A spectrogram is essentially an image, right? So it's 2 dimensions carry meaning, but you chose to flatten the image when you were training your TPM. So I was wondering if we're not overfitting to some spurious relations between pixels in that image, and why you chose to flatten instead of doing something like convolutional neural network or something like that.
Yeah, so I did a lot of searching with choosing what kind of architecture to use. I also use convolutional networks and stuff like that. But I think what seemed really neat to us, and also the reason we chose this model was we wanted to be as biophysically close to what is happening in our brain as possible. So the linear nonlinear transformation seemed closer to how the sensory neurons fire. But you're right, and this also works with convolutional networks, which I've done. But I think what was neat for us that this just a single layer could capture those predictive features, which was kind of neat for to see that. And yeah, you're right that spectrograms is what ideally our brain is not actually listening to spectrograms, right? It's listening to waveforms, and ideally that would be the kind of stimulus that we want to use, but unfortunately, right now, the standard is to use spectrograms for auditory input.
I mean, I think it's good to use spectrograms because there is evidence that the brain cares about these frequency components as well, right? Yeah, but thank you anyway.
Yes.
Cool. In the interest of time, we're going to go to the next talk. Thank you again.
Thank you.
Next, we'll have Emily to talk about model connectivity, overcoming the limitation of functional connectivity by leveraging the power of the VOXELWISE encoding model framework.
That's not important. Let's see. Okay, we'll start that again. Great, okay. Good morning, everyone. My name is Emily Meschke. I'm a graduate student at Berkeley in the Gallant Lab. And today I'll be talking to you about model connectivity. So the earliest human neuroimaging studies focused on mapping functionality onto different brain areas. But more recently, focus has shifted into understanding how these brain areas interact and actually form functional networks. So the most popular method of recovering these functional networks is called functional connectivity. But in this talk, I'll argue that functional connectivity suffers from some fundamental flaws that limit its sensitivity and interpretability. So to address these flaws, we developed model connectivity, which builds on the VOXELWISE encoding model framework. So in this talk, I'll first start by describing the flaws with functional connectivity. Then I'll introduce how model connectivity addresses these flaws. And finally, we'll see how the flaws with functional connectivity manifest when we apply functional connectivity to a narrative auditory data set. So to start, we'll just go ahead and define functional connectivity. So functional connectivity is based on temporal co-fluctuations in bold activity. In this example, we've recorded 3 bold activity from 3 different voxels. And then we can compute the functional connectivity between pairs of voxels by calculating the Pearson's correlation between their time series. So importantly, functional connectivity assumes that these co-fluctuations in bold activity actually indicate that brain areas are functionally coupled. So with this method and this assumption, functional connectivity is often used to define the cortical extent of functional networks, mapping functionality onto different brain areas, and also to assess the degree of functional coupling between areas to quantify how much these areas interact. However, we argue in this talk that both interpretations are confounded by the fundamental flaws with functional connectivity. The first and arguably most insidious of these flaws is that functional connectivity does not differentiate between signal and noise-related fluctuations. To demonstrate the magnitude of this problem, I've plotted temporal SNR and 2 example time series, one from a voxel with high TSNR and one from a voxel with low TSNR. The important thing to note is that temporal SNR is not actually related to function. It's related to differences in signal quality as measured by MRI. And these functionally relevant fluctuations can actually constitute a large portion of the signal that we're recording and thus bias the results of functional connectivity. TSNR also varies across individuals and across runs. Here I've plotted TSNR that's been calculated on individual functional runs from 2 different subjects across 2 different runs. And if you're not familiar with looking at flat maps, just to orient you, this portion is early visual cortex, medial parietal cortex, prefrontal cortex, and then temporal cortex. So if we compare these maps across different functional runs, we can see that, for example, in this portion of prefrontal cortex, the pattern of temporal SNR is very similar within subjects, but very distinct between the 2 subjects. Furthermore, there's this portion of early visual cortex that is very different between runs in the same subject. And I just want to highlight all of this for you, again, because functional connectivity does not differentiate between signal and noise in the bold activity, and so all of these things could be incorrectly attributed to differences in functional connectivity. The second flaw that we point out with functional connectivity is that there's not an easy way to define the functionality of the recovered networks. So say in this example, we performed functional connectivity, or we applied functional connectivity to some functional data as participants performed some task to recover these 3 functional networks, green, dark blue, and light blue. So the ways that we would typically get at the functionality of these networks is to compare the cortical extent of these networks to maps of task activation, or compare the cortical extent to previously defined networks. So if we kind of stare at the top and the bottom maps a bit, we can identify the networks as the cingulo-opercular or salience network for green. Dark blue would be default mode-ish, and then blue would be frontoparietal-ish. However, these are very coarse functional descriptions of all of the networks. And importantly, in a resting state paradigm, there's no way to further infer functionality from the bold time series alone. And we argue that because of these flaws, functional connectivity actually isn't very sensitive to differences across tasks. This phenomena is observed in a few papers, such as Cole et al., as shown here, where the authors applied functional connectivity to data that was collected as participants performed tasks and also while participants were at rests. So if you compare the functional coupling profile of the functional connectivity matrices, you can see that the 2 are very similar. So even though task activations change and the behavior of the subjects change, the patterns of functional coupling as measured by functional connectivity don't actually change. So others have suggested, like Grattan et al., have suggested that this means that functional connectivity is simply not well suited for studying functional differences across tasks, and it's better suited to studying individual characteristics. But we argue that this insensitivity to functional differences is actually reflecting a strong influence of noise in the results of functional connectivity. So to address these problems with functional connectivity and to recover or develop a network, or not a network, develop a method that can recover functional networks that are sensitive to task differences, we developed model connectivity. So on the left, with functional connectivity, you correlate the time series of bold activity between voxels. But with model connectivity, we actually build on the VOXELWISE encoding model paradigm. So in this paradigm, we have our experimental stimulus and our task, and we record bold activity as participants perform the task or watch the stimulus. But importantly, we also annotate features over time that are hypothesized to actually predict variability in the bold signal. To actually quantify how much variability these features predict, we fit our model weights using regularized linear regression to recover a high-dimensional functional tuning vector for each of our individual voxels. And then we can map the functional tuning onto different areas of the brain. Importantly, that's only the first part of the VOXELWISE encoding model paradigm, because the model weights that are fit during the train set are estimated or validated on a separate test set, in which participants listen to a new stimulus as we record new bold activity, annotate new features, and then use the same model weights that were already fit to predict bold activity in the test set. We can then get a metric of prediction accuracy for each voxel to see actually how well the functional tuning described by the model weights generalizes to a new set of stimulus. So to adapt this method to recover functional networks, we essentially group voxels or vertices based on the Euclidean distance between their model weights rather than the correlation between their time series. In this work, we applied both model connectivity and functional connectivity to a narrative auditory data set to demonstrate the advantages of model connectivity. So the data set that we were working with was collected by Huth et al., where 9 participants listened to naturally spoken stories as we recorded their brain activity. The authors annotated these semantic features across time, which essentially represents each spoken word in the stories in terms of how often they co-occur with a set of 1,000 common words. We then, because we wanted to, for our first approach with model connectivity, recover networks that generalize across the group, we projected all of the functional data in the subject average space to the group FS average surface, and then fit our semantic models to each vertex in the FS average surface for each subject. To perform functional connectivity, we then took the average bold activity over time. For model connectivity, we took the average subject weights, and then computed the correlation between bold time series for functional connectivity and the Euclidean distance between the model weights for model connectivity. Lastly, to actually recover the functional networks, we applied hierarchical clustering to these distance matrices to group vertices into different networks. So because we used clustering to recover the functional or the clustering solutions, the number of clusters in our clustering solution actually changes the networks that we recover. So to identify the optimal number of networks for our given data set and our given metric, we use the cross-subject prediction accuracy to select that optimal number. What that essentially means is we took our n-1 group average data, the model weights for model connectivity and bold time series for functional connectivity, and used that data to generate different solutions with different numbers of networks. For each of those networks in each solution, we took the network average model weights and used that to predict the bold activity in the test set. We then compared the network average bold activity in the test set to the actual observed bold activity in the held out subject. And that's how we evaluated each of our clustering solutions. So we did that for each held out subject across all numbers of networks. And that's what's plotted here with prediction accuracy as a function of the number of networks per solution. So the first thing we notice is that the model connectivity solutions across all number of networks and across all held-out subjects are better at predicting bold activity in the held-out subject than functional connectivity. Another interesting thing is if we look at this 20,484 network solution, that solution essentially treats every vertex as its own network, so there's no clustering being performed. And interestingly, functional connectivity, even the best version of functional connectivity, does not actually outperform that non-clustered solution. This indicates that even though we're going through all of this extra work to reduce the spatial dimensionality of our data, we're not actually gaining anything in predictive power. So just to go back to the whole reason as to why we generated these curves, we wanted to be able to select the optimal number of networks across subjects. And to do that, we looked at the early local maxima in these curves. And that turns out to be the 13 network model connectivity solution and the 10 network functional connectivity solution. And that's what we'll be looking at for the rest of this talk. So that's exactly what we've plotted here, the model connectivity solution on the left, functional connectivity on the right. At the top here, these dendrograms represent the similarity between all of the networks, where more similar networks are located closer together and are also colored more similarly. The colors are meaningful within a clustering solution, but are not comparable between the two clustering solutions. We can also project these solutions onto the inflated and flattened FS average surfaces, where each vertex is colored according to its network assignment. So the first thing that kind of jumps out is that the largest network that was recovered by model connectivity, and that what we've colored as cortical curvature, spans all areas of the brain that are not functionally relevant to our task. So again, it's a language processing task. So we would expect what good predictions in auditory cortex, TPJ, and medial parietal cortex, and prefrontal cortex, but not necessarily in visual or motor cortices. In contrast, functional connectivity spans all brain areas regardless of its functional relevance. So this begs the question as to why model connectivity is able to be so spatially localized, and more specifically concentrated on these functionally relevant areas. So to examine that, we just looked at the prediction accuracy of our semantic models without performing any clustering, and that's what I've plotted here. This basically is a map that tells us what brain areas are well predicted by our semantic models. So we can also look at the prediction accuracy of each network, and that's plotted in these bar plots here, where each bar is colored according to its corresponding network, and the error bars indicate the standard error of the mean in prediction accuracy across held-out subjects. So we can see that model connectivity recovers more well-predicting networks than functional connectivity. And importantly, the model connectivity solution also preserves the pattern of predictable signal that we see in the non-clustered prediction accuracy above. For example, these blobs of high prediction accuracy are actually preserved in the model connectivity solution, but not in the functional connectivity solution. And this is good, because we want to be able to recover networks that reflect functionally relevant functionally relevant signal. Another reason that we argue that functional connectivity is not as focused on functionally relevant areas is that it's confounded by spatial differences in SNR. So here, rather than plotting network average prediction accuracy, we plotted network average TSNR. This time, all of the networks are sorted according to the magnitude of the network average TSNR. So when we look at these plots, we can see that there's more variability in network average TSNR for the functional connectivity networks, where these green networks tend to have higher TSNR, and this light blue network jumps out as having low TSNR. And interestingly, this low TSNR network also spans brain areas that are known to suffer from signal dropout, such as inferior and orbitofrontal areas. So it seems like functional connectivity is, in fact, sensitive to these differences in TSNR. And just like the pattern of prediction accuracy was preserved in the model connectivity solution, the pattern of TSNR is somewhat preserved in the functional connectivity solution. And this suggests to us that functional connectivity is at least more confounded by TSNR than model connectivity. So now we can actually move on to how model connectivity addresses the second flaw which is that functional connectivity does not give an easy way to define the functionality of the recovered networks. So we can do that. We can define the functionality with model connectivity by examining the model weights. In this particular data set, as I mentioned earlier, each word spoken in the experiment was represented in terms of how often it co-occurred with a set of 1,000 common words. So then we can map all of the words spoken in the experiment to a two-dimensional space using any dimensionality reduction algorithm. We used UMAP here, and in this semantic space, each point represents a word that was spoken in the experiment. So words that tend to co-occur together in natural language are closer in the semantic space. So then for each network, we can examine the model weights and actually determine which words spoken in the experiment best predicted variability in bold activity. And we can do that again for each network. And that's exactly what we've plotted here. In these plots, each colored point represents one of the top 100 words that best predicted activity in that network, with some example words plotted in larger points. So by examining all of these words, we can kind of interpret the functionality of all of our networks. And we can see that these blue networks seem to represent physical-related semantic concepts. The green ones represent place-related semantic concepts. Red networks represent social-related semantic concepts, and then orange represent discourse-related semantic concepts. So lastly, not only can we actually interpret the functional tuning in individual networks at the group level, we can interpret the function of each network in individual subjects. And that's what I'm showing here. Basically, we determined the top 50 words that best predicted bold activity for each network, but instead of using the group average weights, we used the individual subject weights. So if we look within each of these 4 example networks, we can see that the distribution of points, or the actual words that predict bold activity in that network, is generally consistent across subjects within the same network. To quantify that, we can take the correlation between the model weights for each network for each subject, and that's what's visualized here in this correlation matrix, where the rows and columns are ordered according to subjects and then grouped according to network. The main things to note in this correlation matrix is that the blocks of high correlation along the main diagonal indicate that functional tuning is very consistent within networks across subjects, in all networks except for our noise network. And then we can also see within individual network blocks that within the same subject, model weights from different networks are actually more correlated within subjects than between subjects. So in this presentation, I've discussed some of the flaws with functional connectivity and hopefully convinced you that model connectivity is able to overcome those flaws. The first of those flaws being that functional connectivity does not differentiate between signal and noise-related fluctuations in bold activity. A consequence of this is that functional connectivity recovers spatially coarse functional networks that are confounded by differences in SNR across the brain. In contrast, model connectivity uses an explicit model of what information drives these fluctuations. So model connectivity recovers spatially localized networks that are not confounded by spatial variability in SNR. Second, because there's not an easy way to define the functionality of the recovered networks with functional connectivity. Once you recover the networks, there's not really much more you can do. But with model connectivity, since it's based on profiles of functional tuning as quantified by the model weights, we can actually recover functionally interpretable networks. So I'd like to thank Jack Gallant, my advisor, Matteo, who I've worked very closely with on this project, Tom for developing a Python package called Himalaya that implements a lot of the VOXELWISE modeling stuff, and then the rest of my lab for ideas, feedback, and moral support. And of course, all of you for your attention.
Thanks for the great talk. I'm wondering how the networks you recover with model connectivity compare to if you were to just do clustering or dimensionality reduction directly on the feature by voxel weight matrix?
Dimensionality, as in cluster on the PCs, or?
Or just get the PCs, so without doing the Euclidean. I'm wondering, like, conceptually or empirically, how to think about the extra step of doing finding the voxel by voxel Euclidean distance in feature space.
Yeah. So we wanted to kind of mirror functional connectivity in the sense that we get these groups of voxels that all represent similar semantic concepts. So yeah, there's definitely different methods that you could use to group them. I haven't explored that one in particular, but there's definitely other ways.
Thanks.
Thank you very much. So this was very interesting idea that via this middle, the model layer or component, you sort of average out is not the right word, but sort of get rid of that noise so that because it's repeatable, then you get rid of that noise. So I'm wondering so everything lingers on the appropriateness of this regularized regression model. So if it doesn't fit, presumably you're going to get the same result as you would get from the functional connectivity. So have you done any sort of sensitivity analysis on whether or not, like how does that affect and how to choose a model? Because in this data set, sure, it showed it was appropriate, but there's no guarantee that your regularized regression would fit.
Yeah, no, that's completely accurate. Essentially, we do rely on having well-predicting features. So as I showed you the map of prediction accuracy, the non-clustered version, it does, span a lot of interesting portions of the brain. But if we were basing it on, a feature space that maybe is more localized, you would only kind of get distinctions between functional distinctions within those well-predicted areas. So it is definitely constrained by, the feature space that you're using.
Hi.
Hello.
My question both builds and perhaps responds to the first question that was asked about dimensionality reduction and applying those sorts of techniques to this data, because I'm aware that problems arise when you apply PCA to fMRI data in an effort to reconstruct functional networks, because even though it's useful for resolving an underlying subspace, the eigenvectors it produces can be transformed, and therefore the subspace validity is not changed, but the mappings of that vector back to the brain can completely be changed, sort of screwing with your ability to move back to the brain. So given that issue with dimensionality reduction and trying to do the sort of work you've been talking about, I'm wondering how your modeling approach might address those problems in terms of maintaining validity to the functional networks you're observing, absent of being able to transform those representations, those weights, and therefore messing with what you're actually seeing on the surface.
Sorry, I don't know if I can, maybe we can talk offline a little bit more about what you mean, but
Sure, no worries.
Yeah.
Okay, thank you.
Cool. Thank you, Emily.
Thank you.
Yeah. Next, we'll have Jenelle to tell us about model metamers and how that illuminates the differences between biological and artificial neural networks.
Hi everyone, I'm Jenelle Feather, and today I'd like to present work that was done in collaboration with my PhD advisor Josh McDermott and our collaborators Guillaume Lasserre and Alexander Madre. And you can find more information about all of this on our preprint on bioRxiv. So our sensory systems, such as the auditory system and the visual system, provide the inputs for how we navigate the world. And recently, as many of us are aware, artificial neural networks have emerged as some of our leading models of sensory systems. These networks are often trained to perform tasks such as speech recognition or object classification. And once they're properly optimized, they do these tasks about as well as humans. And because of this, there's widespread interest in considering these convolutional networks as candidate models of the human brain. Now, one critical part of the representational power of these neural networks is their invariance. For instance, a speech recognition network can successfully work on many different voices. Or an object classification network can recognize a dog from many different viewing angles. And in this sense, it becomes invariant to the pose or the location of the dog. Now, it's worth remembering that this is not an easy task. The pixel values that are present within each of these dog images are very, very different. And yet humans, and now neural networks, are able to recognize all of these images as a dog. And in fact, this is exactly what many of these networks are trained to do. And so it's natural to suppose that the success of these models at capturing human behavior and brain responses may in part be due to them capturing invariances that are similar to those of the human perceptual systems. So to directly compare the invariances between artificial neural networks and human observers, we use the idea of metamers. Metamers are two stimuli that are physically different, but that produce the same responses within a system. Classically, the word metamer was first used in the study of color perception. Here it's possible to come up with two light sources that have very different spectral power distributions, but when they're projected onto the responses of photoreceptors in the human eye, these light sources are perceived to be the same color. And this provided the first evidence for the physiological properties of the three types of cones in the human eye, and also is the basis for most image reproduction technologies like your computer screen. Metamers have also been used to study texture perception and visual crowding. In both of these cases, the precise spatial or temporal information is thrown away to create visual or auditory stimuli that look or sound the same as the original under particular conditions. So, motivated by these previous findings, our work highlights that metamers provide an additional test of any model of perception by simply testing whether the invariances of the model are also present in the human perceptual system. So the main idea behind this work is that a good model of human perception will share invariances with humans. Most current neural network models of perception that we have today are trained to do a specific task, and thus they might develop task-specific invariances. And so we extend previous work to class-specific stimuli. To do so, I'll walk through the framework for how we're thinking about the invariances of a system. We can think about these invariances as a subset of all of the possible stimuli. So for a given reference stimulus, there's a set of stimuli that are considered to be the same class to a human observer. For instance, if the reference stimulus is a cat, then there's a set of stimuli in pixel space where humans will consider each of those stimuli to be a cat. And some people might refer to this as a class manifold. Now, we can look at the model in the same way. If the model has a classification decision, which many of our models do, then there's a set of stimuli that the model will consider to be the same class as this reference stimulus. And we can think about metamers in the same space. As said before, metamers are stimuli that are indistinguishable to an observer. So for this given reference stimulus, there's a set of stimuli that are metameric to humans. Importantly, this is always a subset of the class manifold. Because if these metamers are in fact indistinguishable, then that means they always fall within the same class. And similarly, there's a set of stimuli that are metameric for a model. So these stimuli are indistinguishable to the model, and so once again, they'll always be a subset of the class manifold for the model. Now here, I'm just talking about a single reference stimulus, but you can think about these subsets for many different reference stimuli as well. And in this work, we'll focus on stimuli that are metameric for the model. We will generate these model metamers for a given reference stimulus. One model that we'll consider is a neural network that's trained to do a speech recognition task. To get a model metamer, we take the reference stimulus, in this case it's a natural sound, feed it into the model, and measure the activations at a particular model stage. We then take a noise stimulus, feed it into the model, and measure the activations at the same model stage. To generate the metamer, we use typical gradient descent-based optimization to minimize the L2 norm between the model activations for the original sound and the synthetic sound. We modify this input noise until eventually our optimization succeeds and we have a sound that produces nearly the same activations at this given layer of the convolutional network. And because this model is feed-forward, it also will produce the same output response. After we produce these model metamers, participants are presented with a natural stimulus or a model metamer and had to report what class the image or sound belonged to. We presented metamers that had been generated from many stages of our networks, and will plot the proportion correct across many stimuli. The dotted line that I'm showing here is the network proportion correct, which, as we've designed our experiment, will remain at the level of natural image recognition for all of our model metamers. Now, if the participants are able to recognize the model metamers as well as they were able to recognize the original stimulus, then the model invariances will be consistent with human invariances, and that would be shown as this flat blue line here. If, on the other hand, humans cannot recognize the model metamers that had been generated from a particular layer as well as they were able to recognize the original stimulus, then the model has inhuman invariances. And once again, because our networks are feed-forward, information can only be lost from one layer to the next, and so this might look something like this green curve. As the model invariances are built up at these late stages, the metamers that are generated become unrecognizable to human observers. So for the first set of experiments, we studied supervised neural network models. We investigated visual neural networks of perception that were trained on the ImageNet recognition test. We generated metamers from various stages of these networks. And even from this single example, you can see that as we generate metamers from late stages of the network, they start to look unrecognizable. To see if this result held across different neural network architectures, we generated metamers from 5 different visual models, which span different architecture classes and varied in depth. If we simply look at these example model metamers, in all of the model architectures, the late model stages have very few features of the natural image that they were matched to, which is shown on the far left. So we directly tested the human recognition of these model metamers using a 16-way image classification task on Amazon Mechanical Turk. This is based on a task proposed by the Bethke Lab in the paper by Garris et al. In 2018. As expected, because our optimization succeeded, the network performance on the model metamers was comparable to the natural image for all of the metamer generation layers, shown here in the dotted line. However, in all of our tested architectures, the human performance fell close to chance by the final stages of the network, suggesting that the model invariances don't match up with human invariances. We also investigated auditory networks that were trained on a 793-way word recognition test. A 2-second clip is played, and the network reports the word that's present in the middle of a clip. Similar to the visual networks, we generated metamers from the consecutive stages of each of these audio networks. We then tested audio networks using the full 793-way word classification task, once again using Amazon Mechanical Turk. So humans listened to a 2-second sound clip and then had to report the word that was spoken in the middle of the clip. Now, we found the same trend in our auditory networks, where in both of the architectures we tested, the human recognition of model metamers was significantly lower by the final model stages. Now, we can actually listen to some of these model metamers. So for visualization purposes, I'm showing cochleograms of our generated audio metamers. You can think about these as being similar to spectrograms. So first I'll play the original audio, which we used to generate the metamers.
It's a job security program that prevents
And being shown here are the model metamers generated from each consecutive model stage. Early layers are on the left, while deep layers are on the right. Now, this is a model metamer that was generated from the intermediate stage, convolutional layer 3.
the job security program that prevents
So at this stage of the network, the model built up some invariances, which you can hear as a somewhat distorted pitch or background noise, but this metamer is still recognized as speech. In contrast, by the final model stage, the model metamer doesn't sound like speech at all. And this reveals that the network has learned invariances that don't match up with human perception by this final model stage. Okay, so in this first section, we found that supervised neural networks have invariances that are not shared with humans. But one common critique of supervised neural networks is that they require training on many labeled data points. And this learning procedure deviates markedly from learning that occurs in biological systems. So to test whether these unrecognizable model metamers are simply due to the supervised learning signal, we investigated unsupervised models. The unsupervised networks that we tested were all trained with self-supervision. These networks are trained so that multiple views of an image yield similar embedding spaces, and also some of the loss functions explicitly include a component that pushes apart the embeddings that are measured for different images. Now, while we found that there was a slight increase in the recognition for intermediate layers, for instance, in MoCo V2, which is this orange line, generally, we found that these self-supervised networks have fairly unrecognizable model metamers by the late model stages. And so from this result, it seems like the misalignment of invariances is not simply due to the supervision signal. Now, although using this unsupervised objective did not improve recognition of model metamers, we found that a type of training that alleviates adversarial examples yields networks that have more recognizable model metamers. Adversarial examples are images where a small, typically imperceptible perturbation has been added that changes its classification decision. That changes the model's classification decision. That's an important point. So one state-of-the-art way that we can use to reduce adversarial vulnerability of a network is through adversarially robust training. So here, adversarial examples are generated and added into the training data during the training process, so that a new network is obtained which correctly classifies these perturbed images. Now, if we compare the metamers generated from the standard network to metamers that are generated from our adversarially trained network, we see that the adversarially trained model has noticeably different model metamers. These metamers that were generated from the late model stages start to look much more natural and have a lot of features that are present within natural images. Additionally, we see that our participants are better able to recognize the model metamers from these adversarially trained networks. And we also ran a control experiment where we added random rather than adversarial perturbations into the training set, and this type of a model looks very similar to our standard trained model. So from these results, we see that adversarial training improves the human recognition of model metamers. But one point that's worth noting is that this problem is still not fully resolved. There's still some metamers that are classified differently by humans, particularly in the late model stages. Nevertheless, this is a really good first step in terms of improving our models of human perception. So we extend these results into the auditory domain by training auditory models with adversarial perturbations. Similar to the visual models, these models have more recognizable metamers compared to our standard network. So what is changing in the network representations of these adversarially trained networks that result in more human-like invariances? We've gotten some insight into this by looking at how well one model is able to recognize another model's metamers. So using all of the models previously described and a few more that are not included here, we held out one generation model. This is the model that we use to generate all of the model metamers from each model stage. We then take these metamers and use them as input into another model, referred to here as the recognition model. We measured how well that model could recognize the metamers generated from each layer of this recognition model. Essentially, this neural network is being a participant in our experiment. We then measured we then measured the recognition for many other models. So this is all of the other models that are included in our set. And as a summary measure, we average across these recognition models to see how well the metamers at each layer of a given model are recognized by the other models. And we repeat this whole procedure each time holding out one of our models to use as our generation model. So here we'll look at a few of these models, all with the same ResNet 50 architecture. We can see that metamers that were generated from the late stages of this standard supervised model are not recognizable to our other models. Further, the self-supervised model shows a very similar pattern, where it has metamers at the late stages that are not recognizable to other models. So both of these supervised and self-supervised models have idiosyncratic invariances. Interestingly, the adversarially trained network has much more recognizable model metamers compared to the other types. So this suggests that adversarial training is removing some of these model-specific invariances. Also, from this plot, it's striking how similar it looks to many of the experiments that I presented before, shown here on this graph for comparison. So motivated by this, we directly compared the human performance to the recognition model performance for metamers generated from each stage of each generation model. On this scatter plot on the right, each point represents the recognition of metamers obtained from a generation model stage. The model proportion correct is strongly correlated with the human proportion correct, suggesting that when many other models are unable to recognize metamers, humans will also not be able to recognize the model metamers. The same trend held for the tested auditory models, where human proportion correct was well predicted by the model proportion correct. This experiment also points to a future direction where we maybe could use an ensemble of other neural networks as a way to evaluate whether the invariances are similar to those of the human perceptual system. Essentially, we could make model refinement a little more efficient by using an ensemble of models as a stand-in for our human observers. Now, what do all of these results mean in terms of the result that we showed before, where adversarial training is aligning the invariances between humans and models? So once again, we found that this training removes some of these model-specific invariances by looking at the recognition of those model metamers by the other models. But this is actually a little surprising because it contrasts with the typical view of adversarial examples that they're typically shared across models and often thought to be a function of the training data set. Further, not every model manipulation that reduces adversarial vulnerability improves the human recognition of model metamers. And I don't have time to show this today, but for more information on this result, please see our preprint. So it seems like there's actually something special going on with adversarial training that, for reasons that we still do not fully understand, produces this effect in our model invariances. But to help think about this relationship between adversarial examples and metamers, let's go back to the set description that I showed at the beginning of the talk. So perturbation-based adversarial examples are typically considered to be stimuli that are metameric to a reference stimulus for humans, but that are classified differently from the reference stimulus by a model. And as we've seen from the previous experiments, many neural network models have some metamers that are unrecognizable to human observers. So as we change the model architecture or training procedure, we're pushing around the space of model class invariances and the space of model metamers. For instance, we might eliminate adversarial examples in the model so that the model will correctly classify all stimuli that are metameric to human observers. But this test doesn't guarantee that we have eliminated all of the unrecognizable model metamers. Some stimuli may be metameric for the model, but outside of this human class manifold. Similarly, we might find a model where all model metamers are recognized as the same class to human observers, but this type of a model might still have perturbation-based adversarial examples. Now, instead of either of these cases, we might find a model that correctly classifies human metamers and also where all model metamers are a subset of the human classification manifold. This is a sign that the model is well aligned with the invariances in human perception. Looking at it in this way suggests that eliminating adversarial examples will not necessarily make model metamers more recognizable to humans. Adversarial training, nonetheless, seems to result in the scenario shown here, where there's both a reduction in adversarial examples and an increase in human recognizability of the model metamers. So our experiments suggest that this is because the training eliminates model-specific invariances. But it remains to be seen how it achieves this and how this relates to eliminating adversarial examples. So to summarize our results, we showed that model metamers from late stages of supervised audio and visual networks are unrecognizable to humans. Further, this discrepancy is not unique to supervised learning, as unsupervised models also have unrecognizable model metamers. However, it seems like adversarial training results in models that have more recognizable model metamers at these late model stages. And we investigated how these invariances change in the adversarially trained networks by looking at model recognition of other model metamers. Here, we saw that supervised and unsupervised networks have idiosyncratic invariances, but that adversarial training seems to remove these model-specific invariances. So to conclude, I would like to thank my advisor, Josh McDermott, and our collaborators on this project, Guillaume Lasserre and Alexander Madre. Also, thank you to our funding sources, and as said before, for more information, please see our preprint. Thank you.
Hello, hi. Thanks for the great talk. I'm curious as to what the audio metamers of the adversarial model sound like.
Yeah, they start to sound, they're a little noisy. I don't have any examples here, but yeah, find me afterwards and I can play some examples, but they do sound a little bit more like speech. I mean, they're much more recognizable. Yeah.
Thanks for the very interesting talk.
Yeah.
I'm curious, the networks that you selected, particularly for vision, maybe less for audio, their characteristic is that they're discarding spatial information as you move later in the network. Mm-hmm. So I'm wondering if you considered or would consider future work to look at networks that preserve more information about the scene, and whether metamers from those would be more recognizable?
Yeah, I think that's a really interesting question. So one thing that I sort of like about this, this toolkit essentially is that we can start to ask these questions about many different types of models. And so, yeah, if you have a suggestion of like some of those networks that we could potentially use, I'd be, yeah, happy to run it through. The code's available online.
Thank you.
Hi, thanks. I was wondering if there is scope to actually use these model metamers to train the network, whether that could be like an auxiliary approach to say, like, or in addition to like adversarial training?
Yeah, so it's kind of a little weird because the model metamers have the same activations within the network to begin with. And so if you're using some type of a classification decision from the end of the network, that metamer is going to have the same sort of loss that you would get from your natural image to begin with. And so the typical way of doing adversarial training, you couldn't actually sort of like swap in the model metamers in exactly the same way. One thing that I think would be really interesting is potentially to use some of these other models as some evaluation criteria during training so that you could have, like, make sure that the sort of idiosyncratic invariances of the model are being minimized as measured by an ensemble of other neural networks.
Cool, thanks.
Yeah.
Very cool. I'm wondering if you've tried to construct metamers with respect to the outputs of an encoding model that's based on one of these neural networks, and which you could do for various, different brain areas. And if you think that would be more similar to, subjects' perceptions.
Yeah. That's on my list of things to do, but I have not got around to doing it. I think it's a really interesting future direction.
Cool, thank you, Janelle. I think we'll just cut off questions here, and let's welcome John for the next talk.
All right, thanks everyone for coming, and for to the organizers for putting together this great session, and for the opportunity to speak here today about our work on context-dependent decisions. To illustrate the general scope of this project, I'd like to briefly mention an experiment that the Russian psychologist Alfred Yabos conducted in the 1960s. So Yabos recorded participants' eye movements as they were shown paintings and given different types of task instructions. For example, when asked to evaluate the material situation of the family shown in this painting, they would direct their gaze to specific aspects of the image, such as the furniture or the family's clothing. By contrast, when the very same painting was shown along with the instruction to estimate the protagonist's ages, they were looking almost exclusively at their faces while neglecting all the other aspects of the scene. So what these data beautifully convey is at the heart of this project. How we process the world around us is always selective. It's not a veridical representation of what is actually out there, but a highly filtered view that flexibly prioritizes those aspects that are most relevant in the current context. In the lab, this capacity for context-dependent cognition has been studied extensively with very simple paradigms that require instantaneous decisions about different stimulus features. A recurring theme in this line of research has been that control signals that are thought to originate in prefrontal cortex can bias stimulus encoding to minimize interference and emphasize the processing of task-relevant information. Interestingly, different studies have shown that contextual bias signals can bias sensory representations in qualitatively different ways. Some studies have shown that neural patterns that are evoked in different behavioral contexts are separated from one another in neural state space. Other studies have found that neural patterns that are evoked in different contexts are orthogonalized from one another via rotation in neural state space. And yet other studies have found that sensory representations can be compressed along those dimensions that are irrelevant for the decision in the given context. So while each of these coding schemes seems useful in principle to support context-dependent decision-making, we currently don't know what are their unique virtues and under what conditions they will be expressed. In the current study, we wanted to build upon this work and understand the nature of contextual bias signals that support more complex, sequential context-dependent decisions where multiple steps are taken in order to achieve a given goal. To this end, we considered the domain of spatial navigation, where such sequential decisions are ubiquitous. For example, imagine that you are on your way to meet a friend at the pub, but you still need to collect cash along the way. This requires you to plan a route to a final destination, but via a specific subgoal. A different plan may overlap partially, but nonetheless lead to a different destination, for example, when collecting money on the way to the coffee shop. This example illustrates that flexible navigation requires space and goals to be encoded in ways that avoid mutual interference. In this project, we wanted to understand the mechanisms that make this possible and to examine how those mechanisms may bias the brain's representation of allocentric space with a particular focus on the hippocampus. To this end, we developed a task in which subjects are searching for 2 rewards that are placed under boulders in the corners of 4 interconnected rooms. At the end of each trial, participants can earn a bonus that depends on the time needed to find the 2 rewards. Across trials, subjects search for 2 different types of rewards that are cued by different food items that are shown at the start of each trial. Unbeknownst to them, the task contains a hidden structure. The 2 rooms that contain a reward are aligned vertically for one of the reward types and horizontally for the other one. This structure can be used to solve the task more efficiently. After finding the first reward through random exploration, subjects can directly infer the location of the second reward. Here's an example of the task using a trial with horizontal alignment. Let me briefly start this. Of the to-go state, so at the start of the trial, a food item is shown to cue the given task context. The agent then finds the first reward in the same room, subsequently moves to the wrong room and receives negative feedback there. And then finally moves to the correct room to obtain the second reward and complete the trial. So subjects completed this task on 2 separate days. On day 1, subjects completed a behavioral training session where task context was blocked and 1 set of food items was used to cue the vertical and the horizontal context respectively. This plot that you see here shows the probability with which subjects were choosing the correct room that contains the second reward immediately after obtaining the first one. As you can see, subjects were able to pick up on the hidden task structure and to improve their performance and maximize their payoff. On day 2, subjects completed the task while undergoing fMRI and with context varying trial by trial. They completed 6 runs of the task with 3 different sets of reward items. And for each set, 1 item would cue the vertical context and 1 the horizontal context. As you can see, subjects were able to rapidly generalize their knowledge about the covariant structure and to learn new versions of the task more efficiently. To understand the brain mechanisms that support this kind of flexible sequential navigation decisions, we built a computational model. This model used a population of simulated Gaussian place cells to represent the subject's locations in the 4 rooms task environment. We used the empirically observed movement trajectories for each subject as model inputs and read out the population response as subjects were searching for the 2 rewards. This allowed us to compute simulated activation patterns separately for each room and in each context to recover the representational geometry via representational similarity analysis and to visualize it via multidimensional scaling. Without further elaboration, this model simply encodes the location of the 4 rooms as 2 perfectly aligned quadrilateral spatial maps. However, we equipped the model with 3 free parameters based on previously documented coding mechanisms in the hippocampus during context-dependent navigation. The first 2 parameters reflect well-established mechanisms of place cells in the hippocampus. Place cells exhibit tuning for particular locations in space, but previous studies have shown that this tuning is not fixed, and it can change in different ways when the structure of the environment changes. Some studies have documented a phenomenon called rate remapping, whereby the cell's preferred location remains the same, but the average firing rate changes between contexts. Other studies have documented a phenomenon called global remapping, whereby the cell's preferred location randomly changes between contexts. And further studies suggest that neurons in the hippocampus may encode not only the agent's current location in allocentric space, but also the prospective location of navigational goals. For example, in this fMRI study by Brown and colleagues, they showed that the bold signal in the hippocampus during the planning stage of a navigation task contained information about the prospective location of the final goal state, as well as about the location of sub-goals along the way, but not about other landmarks in the task environment that are not part of the planned route. So based on these findings, we equipped our model with 3 free parameters and examined how they would affect the representation of space in our task. Firstly, we allowed rate remapping to occur. That is, the average neurofiring rate of the simulated place cells could vary between vertical and horizontal context. When this parameter increases, the 2 spatial maps reflecting the 4 rooms in each context become separated from one another via a one-dimensional offset that is perpendicular to the 2 dimensions that encode the spatial dimensions of the map, resulting in 2 parallel planes. Secondly, we allowed for global remapping to occur. That is, the proportion of cells that randomly change their preferred spatial location between vertical and horizontal context could vary. When this parameter increases, the 2 spatial maps become orthogonalized via rotation in state space. Finally, we also allowed for prospective coding to occur. That is, place cells would jointly encode the agent's position in the task environment and the location of the prospective location of the navigational goal. A mixture parameter governed the relative weight given to the current and the prospective location. When the weight for the prospective location is increased, the 2 spatial maps become compressed so that the 2 goal locations in each context become more proximal to each other in state space than they are in physical space. To examine which of these predictions are reflected in the data, we examined movement-related activation patterns in the hippocampus with the exact same approach that we used for the simulated data, analyzing pattern similarity as a function of room and context and visualizing the geometry via multidimensional scaling. And here are the results. Patterns in the hippocampus exhibit an intriguing similarity structure that encodes the spatial distribution of the spatial structure of the task and the behavioral context on 2 factorized axes. Within each context, the similarity of patterns reproduces the quadrilateral structure of the task environment in a low-dimensional subspace akin to a plane. The 2 planes from the vertical and the horizontal context are separated by a one-dimensional offset that is perpendicular to the spatial dimensions of the task, consistent with the model predictions under the assumption of rate remapping. Importantly, the distances between the rooms within each context are not equivalent and do not reflect their veridical distance in space. As you can hopefully see on the MDS plot, the 2 planes are compressed along different axes within the vertical context, primarily separating east from west, and the horizontal context, primarily separating north from south. This corresponds directly to the model prediction under the assumption of prospective coding, whereby goal locations will cluster together in space. We verified these effects statistically by fitting model planes to the empirically observed data of each subject. These planes have 2 free parameters, one for the amount of separation and one for the amount of compression, and both of these parameters are significant in the hippocampus. We then tested the relevance of these contextual bias signals for task performance by correlating them with differences in navigation performance across individuals. And interestingly, we found that the amount of compression significantly correlated with performance, whereas the amount of separation did not. It is also worth noting that the compression of spatial representations is unique to the hippocampus and is not observed elsewhere in the brain. For example, in visual cortex, we find high-dimensional representations of space with very strong separation between the rooms, but very little sensitivity to context or the quadrilateral map of the task space. And activity in parietal and orbitofrontal cortex encodes separated spatial maps, but without compression and without discernible links to task performance. Lastly, to test the presence of prospective signals in the hippocampus more directly, we also examined brain activity at the time when the first reward feedback was received. We tried to decode in which of the 2 neighboring rooms the second reward would be located. This analysis was done separately for each of the 4 rooms, and it aimed to reveal prospective signals encoding the location of the to-be-harvested second reward. And as you can see in the plot here, goal locations could be decoded from feedback-related activity in the hippocampus, but none of the other ROIs that we examined, corroborating the presence of prospective mechanisms. So, in conclusion, our study shows that humans can learn and use abstract representations of latent task structure to flexibly guide sequential context-dependent decisions. Our model-based analysis of representational geometry provides mechanistic insights into the control mechanisms that underpin these types of decisions. Spatial representations in the hippocampus were flexibly distorted to emphasize the dimension predictive of reward in each context, suggestive of dual functions of place cells in coding for the current and for prospective locations. More generally, our findings show that compression of irrelevant information, which is often referred to as adaptive gain control, reflects a versatile control mechanism that supports not only instantaneous but also sequential context-based decisions. So with this, I would like to conclude and thank my many collaborators without whom none of this would have been possible, and also our funding bodies for their generous support. I would also like to share that I've recently started a faculty position at the University of Birmingham and that I will soon be hiring a postdoc for my forthcoming lab. So if you find this work interesting and would like to engage, or if you know anyone suitable, please get in touch or spread the word. And last but not least, many thanks to all of you for your attention.
Hi, thank you for this amazing talk. Beautiful results, meticulous work, it's fantastic. I was wondering where along the long axis of the hippocampus you find most of those patterns, and what you think would be different in those representations, as well as perhaps representations in not orbitofrontal, but a little bit further up medial prefrontal regions, if you had larger sort of set of state space for your tasks.
Mm-hmm. Yeah, it's an interesting question. I've also heard that at the poster session. So far, we haven't really explored it yet, like where exactly in the hippocampus kind of like these compression weights are strongest. We've only done like some basic sanity checks that we can kind of like find it in each hemisphere. I don't know, would you have a particular prediction in this case? What?
Since it's context-based, I wonder whether it's not very posterior, if that makes sense. And so I would expect a little more medial to anterior, and in case mid to anterior, and then in case you have a larger state space, I would expect it to move further anterior and more be reflected in the PFC. But I would absolutely love to see it, and also a lot of your results are consistent with our modeling, so this is exciting. If at some point the data is available online, I would love to try some of those models on it.
Yeah, absolutely. Happy to chat more.
Thank you.
Thank you.
Cool. So I guess since we don't have any more questions, we will just end here. Let's thank all the speakers for their great talks again. And we'll come back at 11:45 for the next contributed talk session.